Yes, We'll start in a couple of minutes. Yeah, good evening. So now we'll start with the sample questions for the course Metabolic Engineering, Week 7. I hope my screen is visible, Ignatius.
Oh, yes, absolutely.
Yeah, okay. Um, yeah, before starting the general announcement, so next week we'll have one class on Monday, which will be to clarify doubts, if any, for the content lectures of week 8. And apart from that, we'll be having another class mostly on Tuesday, again from 6 to 8, which will be a revision session where the entire contents of all the lectures of all the 8 weeks will be like summarized. And if you have any particular doubts or questions, you can inform me in prior so that I will have the necessary preparation to answer that questions. You can post your queries if you have any in the comment box. I'll make a note of them and I will focus the discussion on your doubts specifically in case if you have any. Uh, So we'll start with the questions. So the first question is, compared to other genome editing methods which use selection markers, CRISPR-Cas enables the genome editing under in vitro condition. And the question is true or false. You can take a couple of minutes and try to then answer this question. Yeah, hi Surendra, we just started. Before starting, we just in the first question.
Okay.
So just another announcement. In next week will be week next Monday we'll have the same tutorial session for week 8's lectures. Apart from which we'll be having one more session mostly on Tuesday again for 2 hours from 6 to 8, where we'll be reviewing all the contents of all the 8 weeks. So if you have any specific doubts or questions that needs to be addressed, you can inform in prior so that we'll have the necessary slides or whatever has to be discussed and we can focus the discussion on that particular topics.
Okay, okay.
Yeah. So we just started with the first question. The question is, compared to other genome editing methods which use selection markers, CRISPR-Cas enables the genome editing under in vitro conditions. The options are true or false. You can take a couple of minutes and then try to come up with a rationale as to whether this is true or false. Yeah, who would want to go first? They can first try to tell the rationale and then we can discuss the answer. Hmm. Ignatius?
Yes, sir.
Yeah,
So, first like to put forward my views and then I could maybe tell whether it's true or false.
yeah.
Compared to the other methods of genome editing, I know for a fact that CRISPR-Cas does not use selection markers. It's not
Hmm.
necessary.
Not
But
necessary.
I'm
Yeah.
not really sure if it is applicable only for in vitro or in vivo conditions as well. So,
Hmm.
yeah. So, with respect to the selection markers, the answer would be true.
Sorry, that was not very clear. Audio was not
Yeah.
very clear.
Okay. With respect to the selection marker part,
Yeah.
so the answer would be true because they don't necessarily use selection marker.
Hmm.
But whether it's in vitro or in vivo, that's my confusion.
Okay.
Yeah. So I would go for true only.
So you would choose true. Okay.
True. Yes.
Okay. Huh. So the you although they've talked about selection marker, what they're specifically asking is whether it can be done in vitro conditions or not. We'll listen to Surender's rationale and then we'll discuss the answer further.
Okay. So, in my opinion,
Yeah,
it is false.
it's false. Hmm.
It's false.
And the reason being, like,
Because here the editing are done inside the cell. We put the plasmid DNA
hmm,
containing the guide RNA.
hmm,
Okay, if Cas is already present inside the cell,
hmm,
Cas protein.
hmm,
Okay, and if we want to do the homology directory recombination,
right.
we have to put the desired sequence in another vector.
Hmm,
So everything is done inside the cell. That's
hmm,
why it is false statement. No?
right. Yeah, yeah. So to rephrase what you said, uh, So, okay, I'll first summarize how CRISPR-Cas works. So, in CRISPR-Cas, we have a guide RNA that, with the help of nuclease, which is Cas, CRISPR-associated proteins, what it does is it goes and binds to a specific sequence and it cleaves there. But after this cleavage, what is important is the double strand break has to be repaired, and there are multiple methods that can be done in vivo. But this double strand repair is very difficult to achieve in vitro, or even if the necessary enzymes are provided in the test tube. So the right answer is that it's false. It can only be done in vivo conditions. And Ignatius, to answer, so you were stating that CRISPR-Cas can be done in vitro conditions, right?
No, sir, I was
You
confused
were confused.
with
Okay.
that part. Yeah.
Okay. So, although we can provide the Cas protein and the guide RNA and the genomic DNA within a test tube, and although the guide RNA can guide the Cas protein to the specific site to cause the cleavage, but the double strand break, it is difficult to repair in vitro. But even if this double strand break is repaired in vitro, the entire genomic DNA cannot be inserted into a cell. So, due to these limitations, we can't really say that CRISPR-Cas can perform genome editing in vitro conditions. So, we just stick on to that CRISPR-Cas does genome editing in vivo conditions. Was that clear?
Yes, sir.
Yeah. Okay. Huh. The next question is, what does Cas do in CRISPR-Cas gene editing technology? And there are four answers. Tunable regions, B is molecular scissors, C is transcriptional modulator, option D is BNC, and option E is none of the above. You can take a couple of minutes and then try to come up with the right answer. Yeah, Surendra.
Cas is a endonuclease,
Yeah.
so it is a molecular seizure,
Okay.
and it cuts the DNA
Huh.
that is complementary to the guide DNA.
Hmm. So as the
It makes the double stranded.
as the guide DNA interacts in hydrogen bonds to its complementary guide, as the guide RNA, it's an
Then
RNA. As the guide RNA
uh,
interacts with the complementary DNA, the position is
uh,
fixed, then it does the cutting. Yeah.
so it is changing the gene expression
Huh.
at DNA level. Okay,
Huh. Huh.
means it is breaking the DNA.
Hmm.
So transcriptional modulator, it is not transcriptional modulator. That's why, because there is no transcription happening here in that case.
Huh. Okay. We'll discuss that further.
Okay, okay.
Okay. So you're
So in
saying
my
it's
opinion,
option
the
B. Okay.
yeah,
Right. Okay. Ignatius, do you go with what Surendra
Yes,
said?
sir.
Or
No, sir. My answer would actually be option D,
huh?
both B and C,
Right.
because when we're talking about CRISPR-Cas9 as a gene editing technology, there are two aspects to it.
Hmm.
So one is your interference,
Hmm.
and one is your activation,
Hmm.
CRISPR activation.
Right.
So either your Cas should be conjugated to the transcriptional factor or a promoter region, and then could be introduced into a particular site in order to
Hmm.
produce a particular protein contained product.
Hmm.
So I think apart from the molecular cellular aspect, I think they are even transcriptional modulators. So I think B and C will be correct.
Right. Okay. Hmm. Okay. Yeah. So now to rephrase what you said, like how Surendra had mentioned the role of Cas protein as a molecular scissor, where with the help of guide RNA, it is able to locate a specific gene of interest, go there, sit on the DNA with on the gene of interest, and cut it. That is one role that it plays as a molecular scissor. And the second option that they're talking about is transcriptional modulator. They don't specifically say whether it is activating or repressing. So they just say that it's modulating the transcriptional level. And as we've seen in the last lecture of week 7, there are two kinds of behaviors that can be expected depending on the kind of adaptations or fusions that are being done to this CRISPR and Cas independent, the guide RNA and then endonuclease. So one is a method where we have RNA interference, where uh, the expression of a particular gene, it can be reduced. So what specifically happens is you have uh, deactivated Cas9, meaning it has two mutations that are responsible for the nuclease activity. Now, because of these two mutations, their nuclease activity is lost. But still, this deactivated Cas9, it can bind to guide RNA and it can sit on a complementary DNA. So now what happens if there is an obstacle in front of an obstacle on the DNA? This would prevent an RNA polymerase from transcribing. So the transcription is stalled. So this is one method in which transcription can be downregulated. Modulation is downregulation is a part of modulation. The other method is upregulation. In upregulation, what they do is they fuse this deactivated Cas9 to one subunit of the RNA polymerase complex. Now, due to some conformational changes, this acts as a transcriptional activation domain, and further, act, active Cas9 or how do I say? Upon a specific signal, this is released and transcription happens, enabling upregulation of specific genes. So these are two ways in which transcriptional modulation can be achieved using the CRISPR-Cas technology. So what exactly that answers this question is option D, which is BNC, molecular scissors and transcriptional modulator. Was that clear, Surender?
Yeah, clear.
Right. Okay.
So we can change the promoter also, na, by this technique.
Yeah, so what they do is with respect to RNA interference, they choose the guide RNA such that it has some region close to or nearby the promoter, so that correctly when RNA polymerase sits on it, it cannot move, it cannot even start transcription. That is how RNA interference is achieved.
Mm.
Yeah.
Okay.
Huh.
Got it.
Understood. Okay. We'll go to the next question. In reconstructed CRISPR-Cas9 system from Streptococcus pyogenes, the endonuclease is option A, a fusion consisting of only the Cas9 nuclease and a single guide RNA containing the essential CRISPR RNA and the tracer RNA elements. Option B, a result of fusion between PAM and guide RNA forming two component endonuclease system. Option C, a fusion where Cas9 protein only requires a seed sequence within this CRISPR RNA and a conserved protospacer adjacent motif upstream of the CRISPR RNA binding site. And option D, a result of fusion between PAM gRNA and crRNA. So we can take a couple of minutes and then try to answer this question. Yeah, does anyone would want to attempt this? Surinder or Nishis?
So, uh, can I explain?
Yeah, yeah, sure.
So, for CRISPR-9 technology,
CRISPR
CRISPR-Cas9,
Cas9, yeah.
time sequence should be in the targeted DNA.
Yeah, PAM is naturally present in the genomic DNA. Okay.
Genomic DNA that we
Yeah,
have to target, that we are targeting.
yeah. PAM sequence
Okay.
is what our guide RNA plus the tracer RNA will be able to, I mean, the tracer RNA, which is a part of the guide RNA, and the cRNA, which is having the repeats, will be able to locate. So PAM is present in the genomic DNA. Yes.
Hmm. Yeah, genomic DNA.
Yeah. Yeah. You can go ahead. Yeah.
Hmm. And TAM, it should be downstream. It should be downstream. The proto sequence, not
Uh,
upstream.
yeah. So okay, it depends
TAM,
on
TAM should
it.
be downstream.
It depends on how you are able to say with respect to the genomic DNA. You're saying is it downstream of genomic DNA or upstream of genomic DNA? It's up to how you say that.
Downstream of the sequence.
Okay. Yeah. So protospacer is the one with which our CRISPR
Guide
RNA
RNA will bind.
interacts. Yeah. CRISPR RNA. So the guide RNA,
Guide RNA will bind.
uh,
Uh,
huh,
so, correct answer is A.
option A. Okay.
Yeah, fusion of the Cas9 guide RNA and the CRISPR RNA and tracer RNA element.
Right. Yeah, but before we go
Because
on
TAM,
to, yeah,
TAM should be in the genomic DNA.
PAM is in
It
the
is not the
genomic
part of
DNA.
the. It is
True,
not the part of the Cas9 system.
true,
CRISPR Cas9.
true, true.
Okay.
Huh. But before going on to the answer, would you want to explain what CRISPR RNA and tracer RNA are?
CRISPR RNA actually, it is a guide RNA.
Okay, so they are in the
Guide.
option they are saying a single guide RNA containing the essential CRISPR RNA. CRISPR RNA is cRNA and the tracer RNA. Tracer RNA is transactivating CRISPR RNA.
Transactivating, yeah.
Yeah. So, like, do you want to explain what CRISPR RNA and transactivating CRISPR RNA is?
CRISPR RNA is exactly complementary to the protospacer sequence.
Right.
That
So,
is
do you,
exactly complementary.
huh? Okay, that is CRISPR RNA. Okay.
And transactivating RNA element, they activate the Cas9 if PAM sequence is there. If they find the PAM sequence in the genomic DNA.
Right. 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 So, another feature is that we have not yet discussed is this tracer RNA also has like stem loops and it is nearby the RNA which is complementary to the CRISPR RNA. So what it looks like is you have CRISPR RNA. To this, you have the tracer RNA, which has stem loops. And this is the 5 prime, this is the 3 prime, and the entire of this is inside Cas9. So this specific single stranded region is the cRNA, whereas this is the tracer RNA.
So guide RNA is made up of the CRISPR RNA and the
Tracer RNA.
trans-activating RNA.
Yeah.
And
So
CRISPR RNA is exactly complementary to the protospacer.
yeah. So CRISPR RNA has a sequence called the spacer. So within cells, how it is arranged is you have a sequence that is taken from an invading virus. What happened? Okay, yeah. Huh. So within the cell, you have a sequence from the invading virus that is called the spacer. And next to that, there is something called as a repeat that is unique. So you have this unique repeat followed by a spacer and multiple like that. So that spacer is the CRISPR RNA. And the region complementary to the spacer is called the protospacer. So this interacts with the protospacer. Let me draw it in a different color so that it's clear. Yeah, so you'll have the protospacer here. And downstream of this, we'll have something. This is the PAM motif. Yeah. So this is how the entire of, I hope it's slightly better now. So we have the CRISPR RNA, the tracer RNA, the protospacer, and the protospacer adjacent motif. Now, to answer the question, in Streptococcus pyogenes, the endonuclease, which is this red color box, which is the Cas9, it contains a fusion of the Cas9 nuclease, this single guide RNA, which has both the CRISPR RNA and the tracer RNA elements, which is option A. Uh, was that clear, Ignatius?
Yes, sir.
Okay. Yeah. Yeah. So the next question is CRISPR-Cas system is most abundant within option A archaeal domain, option B bacterial domain, option C eukaryotic domain, and option D all of the above. Uh, we can take a couple of minutes and then answer this question.
So can we start? It's a memory based question.
Right. If there's nothing to think, we can start. Sure.
It is memory based question. Na?
Yeah, yeah, yeah. There's nothing to think. True.
So archaeal domain having the most abundant CRISPR Cas.
Okay, they're not asking abundance. They're asking in which of it is
Abundant. Okay, okay,
abandoned.
okay,
Uh,
okay,
like
okay.
abandoned as in it is not present.
Okay, eukaryotic domain.
Right. Hmm. Do you have any other or difference of opinion, Ignatius? Yeah. So CRISPR-Cas is available or observed in both bacterial domain and archaeal domain. as an adaptive immune response, but this is not specifically seen in eukaryotic domain, although it can be used in eukaryotic domain for genome editing purposes. Yeah. The next question is, in CRISPR locus, interspersed spacers are of A. Uniform length and identical sequences. B. Uniform length and variable sequences. C. Variable length and identical sequences. D. Variable length and variable sequences. So, yeah, we can take a couple of minutes and then answer this question. Hmm.
So these interspace spacers, these spacers actually derive from the pathogens
Yeah.
like
Hmm.
viruses that infect the bacteria.
Right.
From one infection, they get a particular spacer sequence.
Yes. From
If there is a 32,
one particular organism, they take a specific sequence,
that is
and
called the spacer.
that is called a spacer. True.
If there are the 35 spacers, it means 35 different kind of the viruses have infected the virus, infected the bacteria.
True. No, I mean
So
it has been passed on. It doesn't necessarily mean that that particular bacteria has been infected. It has been passed on. So the entire generation of bacteria previously have undergone those infections and they have passed it as a memory in their genome.
yeah, yeah, means they have encountered the 35
Yeah,
viruses.
yeah,
So these spacers are of not same sequence. It indicates they are of not same sequence.
yeah. They are variable
Variable
sequences.
variable sequences.
True.
If we talk about their length,
Hmm.
length also varies. But
Hmm.
if we take the average, it comes 32, 32
You
base pair. If we
uh
take the average,
okay. Should that always be the case?
ideally it is variable. Spacer sequence length also varies
Right.
from uh, means 20 to 70 base pair. Exactly, I'm not remembering the figure.
Ha ha.
But if you take the average, it comes to the near to 35 base pair. If we can say 35 uniform. So in my opinion, uh, length is also variable. Variable length and variable sequences. I will
Okay.
prefer this answer.
Variable length and variable sequences. Okay. Okay. Right. Huh. Ignatius?
Yeah.
Yes, sir. So when it comes to the array that we are talking about, so we have our repeats and we have our spacers,
Right.
and so our spacers are said to be unique. So when we talk about as well as the months,
Wait. Your audio is not very clear. Can you repeat again?
Yes, sir.
Yeah,
So like the spaces are said to be unique sequences
right,
as compared to our repeats that are
right,
present.
right.
Right? So that automatically implies that the sequences are variable and the lengths will also be variable as different viruses could probably have, pass on different lengths of the DNA or RNA to the bacterial genome.
Hmm, hmm, hmm.
So I feel D should be the option.
Right. Yeah. Yeah, both of you prefer to say that the answer is D, variable sequences and variable length. Just a correction, Surinder. So you had mentioned that the average length is 32 base pairs.
Mhm.
That was also mentioned in the slide, but that is for the repeat sequence. So each of these spacers are flanked on both the sides by some sequence that is specific
Repeat.
to a particular bacteria.
Yeah.
One particular bacteria has a set of sequence on both sides of the spacer. It's called the repeat sequence. In the
Mhm.
CRISPR array, this is there. So in the slides, they had given that this repeat sequence averages about 32 base pairs, but no
Okay.
specific length was given, in my knowledge, uh, to the length of the spacer sequence.
So what is the length of spacer sequence?
Spacers, I don't really think that it was mentioned, but for repeat sequences, it was mentioned that the average was 32 base pairs. Maybe I can check and let you know.
So it is also said by the professor, repeats
It
and
could
spacer
be,
are of identical length, same length more or less. They are having the same length.
oh, is it? I don't know if I missed it, but
Okay.
uh, but what I am aware is uh, every in a particular bacteria, all the repeats have the same length and same sequence, identical sequence, uniform length, repeats. But
Mhm.
spacers have variable sequences. For the
Okay.
lengths,
Variable
I
sequence.
variable sequences, and I believe the length is also variable. But let's
Okay.
just check the
Got
answer.
it.
Yeah,
Mhm.
okay, but it's mentioned as uniform length and variable sequences. I need to check on this. Let me make a note of this and let me check on this.
Okay.
Because in my knowledge, the length need not be specifically conserved upon different infections. So let me check on this. Variable uniform length. This is,
Because now one more point I
yeah,
want to share here.
yeah,
Because enzymes are
sure.
very specific. Enzymes are very specific. Okay. If any viruses they are putting their DNA inside the bacteria, it is first infection. Okay.
Right,
Any virus that is putting its DNA inside the bacteria,
right.
so Cas will cut first. Cas will cut first
Okay.
and find particular sequence in
There
the
are many,
CRISPR.
yeah, there are many cas. I think here cas 1 and cas 2 will cut.
Yeah,
Yeah,
Cas1, Cas2.
yeah.
So enzymes are very specific. So I think length should be also uniform
No, the, okay, so,
because they recognize a particular sequence and then cut.
yeah, and it, so the, what we need to understand is when we have a genomic material of a virus, and when these Cas proteins bind to two ends of, when they recognize specific sequences on the genomic DNA, it need not always be uh, around the same length. Like, for example, if you can see my hands, let's say this is one Cas 1 and 2, this is one Cas 1 and 2. Let's say this is the sequence this that they recognize. In one organism, the interdistance between the two recognition could be this much. In another organism, it could be this much. So we don't really know if that is conserved.
Okay.
And th-
So
yeah,
that is if you're talking with respect to two different bacteria, right?
I couldn't hear you. I could hear the different bacteria, but can you repeat again?
Voice is not clear. Ignatius.
No, like you told the difference, right?
Can you come again?
Like you told that it would differ in different species, right?
of so
Is
let's say
that
you have
of
different
bacteria or are we?
no, no. So what I'm saying is CAS, like what Surendra had mentioned, CAS has specific recognition sites that it would bind to only specific recognition sites. What I was telling is within among different viruses, the distance between the recognition sites could be variable, giving rise to different variable lengths.
Right, right, got it, sir.
Yeah. So even I was under the opinion that it could be variable length and variable sequences, but let me check on this.
Okay.
Yeah.
It is also mentioned in one research paper.
Sorry?
Most CRISPR spacer are either 30 nucleotide or 32, 35 nucleotide in length.
In which paper is it?
It is the title is Frontiers of Genetics.
Okay.
Frontiers in Genetics. The title is Investigating CRISPR spacer targets and their impact on genomic diversification of Streptococcus mutants. I'm putting here in my message. No?
Huh.
This one.
Yeah, we've
This one.
got the paper. Huh. Yeah, most CRISPR spacers were either 30 nucleotide or 32 to 35 nucleotide in length.
So,
Right. Hmm.
as I already mentioned, na, professor mentioned in the PPTs
Right.
that repeats and spacer are having the same length.
Okay. So the average 32 and this 32.
Average 32. That's why they said more or
Right.
less same length.
Right. Right. I might have missed it. I'll check that. Thanks.
So meanwhile, we are considering the answer as a uniform length and variable sequence.
Yeah, that's the answer that is given by the course content. Uh,
Okay. Okay.
yeah. Huh. Yeah. So the next question is, which is false for a leader sequence in CRISPR-Cas system? Option A, it has limited base pairs. Option B, it lacks coding potential. Option C, it is always found on one side of the CRISPR in a fixed orientation. Option D, it is 80% identical within a genome but quite dissimilar among species. So we can take a couple of minutes and then try to answer this question. Yeah, does anyone want to start by mentioning what a leader sequence is and then going on to rationalizing which is the right answer or the wrong answer?
This leader sequence they are present upstream of the CRISPR,
Huh,
upstream of
right,
the CRISPR,
right.
and they are necessary for the transcription of the CRISPR
Now,
to make the.
when you say CRISPR, it could mean a lot of things. So let's say whether CRISPR is a repeat sequence or a spacer sequence or the gene that codes for the Cas proteins. So let's clarify on that. When you say upstream
Okay.
of CRISPR, so like, would you want to clarify on that?
Yeah, yeah.
Yeah.
So I'm considering the CRISPR as a repeat spacer, repeat spacer.
Okay. Clear.
Okay,
Right. Right.
I mean spacer. I mean CRISPR. It is
Right.
repeat
That is
spacer.
CRISPR. Okay.
Okay.
Fine.
So this leader sequence they are upstream of the CRISPR,
Okay.
and they are necessary for making the cRNA,
CRISPR
CRISPR
RNA.
RNA.
Right.
So they are having the promoter features, promoter like properties. Okay,
Okay.
and they also have the limited base pair because they are having approximately 200 base pair only. Okay,
Okay.
so rest is the 80% identical within the genome.
Hmm.
So I am having this confusion because all first three options are seems correct with for me. They don't have coding potential,
Right.
and they are promoters. They are behaving like a promoter, so they are present in a fixed orientation. Their orientation cannot be changed.
Right. It there is
So
okay. As far as I remember, there was no mention of the leader sequences acting as promoters, but the leader sequences are like structural features that are near this repeat spacer region. So this can be imagined in a manner similar to leadered mRNA, which take up a particular structure and aid in transcriptional activation, transcriptional modulation.
that's why
So
I'm saying promoter like features.
yeah, but there was no mention of whether leader acts as the promoter or not.
Yeah,
So
exactly.
let's not try to say that it is promoter like, because when you say it is promoter like, it means that the transcription start site could be there. Whereas this leader itself is being transcribed. So we don't really, it was not clearly discussed in the lecture whether it has the transcription start site or not. So let's not say that it's a promoter. But what else was discussed? Uh, Ignatius?
Yes, sir.
Yeah.
Uh, so the fact, then I'm confused with the option C really, sir.
You're confused with option C.
Yeah, option C.
Option C. Oh, what is the confusion with option C?
So is it found like on one side of the CRISPR array? First of all, like, does it act as a promoter or is it a sequence that is being transcribed?
It is being
Leader
transcribed.
sequence.
It is being transcribed for sure. So what happens
Okay.
is you have uh, you have the leader before which you have the Cas genes, and after this leader you have the spacers and repeats and spacers and repeats, and this goes on. So this is gas. So These are transcribed. This is also transcribed. This is also transcribed. All of these are transcribed. What was mentioned in the lectures is that this is transcribed but not translated. So are these. None of these. Only, okay, translation stops with the Cas genes. Only the Cas genes are translated. Whereas this is not being translated and this is not being translated. So we can say that it lacks a coding potential. And secondly, this is usually present within the Cas genes and the repeat sequence in a fixed orientation. That's why we can consider that the answer always found on one side of the CRISPR in a fixed orientation to be right. Now the next question is option D, which is 80% identical within a genome but quite dissimilar among species. Is this a specifically true statement? And also, what has been described about the length of the leader sequence? So, if there is any idea on the length or the conservation pattern, we can go ahead with it. Surendra?
Since it's being transcribed now, so I think option A also should actually hold. I mean, it should be false.
Uh,
Uh-huh.
yeah, yeah, Surendra, 1 minute. Yeah, can you
Uh-huh.
repeat? It was not very clear. I think there's an issue with the mic.
Okay.
Yes. So since like the leader sequence is also being transcribed,
Yeah,
so I think it would. I mean, it's not limited to a specific small number of base pairs. I think it could have like a larger sequence as well.
okay, okay. Huh, right. Surendra, so we were
Mhm.
discussing that options B and C, which is it lacking a coding potential and is always found on one side of the CRISPR in a fixed orientation, to be
Yeah.
right for the leader
Yeah.
sequence. And we were
Yeah.
next
Yeah.
discussing whether it has limited number of base pairs or if it's identical within the genome. So if you have some idea about the length of the sequence or the
Length is
conservation pattern,
limited. They are 100 to 300 base pair.
yeah, a few hundred base pairs. Yeah,
Few hundred base pair.
few hundred base pairs.
Yeah.
Huh. And
So length is
regard
also limited.
and regarding the conservation within
Huh,
genome,
it is. I am having doubt. So I choose this option.
you're choosing option D. Okay.
Huh.
Huh. Ignatius, would you want to go with D or A?
I think I would go with this.
A. Okay. Oh, right. As far as I know, because this is a structural RNA, the leader sequence, I think the option that it has limited base pairs uh, doesn't really convey the meaning of a structural RNA. So even I think it could be limited base pairs. Let's see. So they're saying that the answer is option B, option A, that it has a limited base pairs. Right,
Okay.
right. So before we move on to the next question, I'll just give an example of what a leadered RNA is, leadered mRNA. So you might have read this in gene expression, where a specific RNA might even have riboswitch, like it can bind to uh, metabolites and alter gene expression. So these sequences in front of the other RNA take some structural role and it also plays the role in the expression of downstream genes. Hmm. There's no right. There's no clear explanation, but leader sequence. Okay. This might explain what I wanted to cover. So here they say that the leader RNA of magnesium transporter gene MGTA senses both Mg2 plus and proline. So the ribosome translates the complete MGTL. When low Mg2 plus is there, it forms a non-state because of which the stop codon is not available for ribosome and the ribosome stalls. But when there is high proline then and high magnesium, it is the stop sequence is available because of which ribosome can translate the complete MGTL. So it basically plays a structural role and that interacts with other components in the environment, which means that it cannot be of smaller length. It need to have hundreds of base pairs for it to achieve the structural role. Right. Was that clear, Surendra?
Sure, but I had a doubt.
Yeah.
Yeah, but so the role here basically is it acts as a transcription terminator, right?
Here in the example
It
that we
isn't
were
being
seeing,
transcribed here.
it's a conditional repressor. Yeah, you can it doesn't involve in transcription but rather translation. What it prevents is a place for ribosome to move. So the ribosome has to move across your translated RNA transcript, which is the RNA, so that it can form the protein. But in this specific case, where there is low proline, this takes up a specific structure involving the stop codon. So the ribosome is stalled here. But at high proline levels, it is not stalled. And translation happens because ribosome can move across it. Now you can like ask your question again and we'll see how this works. Hi Ignatius.
Yes, sure. So my
So,
doubt was, we were told that the leader sequence would be transcribed, right?
Leader sequence is transcribed. So whatever is shown here is an RNA only. Transcription is where DNA is
Okay,
converted to RNA. Yeah,
okay, got it.
yeah. Once you are transcribed, you get the
So it
RNA
solves the
as
translation process.
yeah. In this particular case, we have an example where translation is affected. Yeah,
Yeah, okay, sir, got it.
right. Yeah, Surendra.
so it is mentioned in this paper also. Leader sequences they are helpful in the localization of the spacer in the CRISPR sequences.
Hmm.
They are conserved. It is written in this paper also.
Okay.
In the abstract, it is written integral part of the localization. So it is conserved regions of CRISPR.
So what they say is among the kind of leaders that they took, they found a pattern that has been conserved and has been beneficial for as a localization signal. And
Yeah.
what is given here is it's 80% identical within genome but dissimilar among species. So although they talk about conservation, it that this particular paper cannot be like directly used to state that
Mhm.
option
D.
D. Yeah. So
So this
yeah.
paper indicates that you cannot change the sequence, later sequence.
No, what
You
this
cannot.
paper indicates is
We
if you
cannot change
change
later
the
sequence.
if you reduce the length of the uh,
Yeah.
leader sequence, which is mentioned by truncation of the leader. If you reduce
Yeah.
the length, then the spacer acquisition level uh,
Reduce.
is reduced. So they don't specify uh, anything beyond that regarding
Mhm.
the conservation. So we cannot use that to say that
Okay.
about the identity. But if there are any papers and if you think that uh, that about the leader sequence, you can share that. We can have a look at that as well.
Okay.
All right.
So its correct answer is A,
Yeah, the
limited
correct
sequence.
answer is A. It cannot have limited base pairs. Yeah. Huh. So the next question is identify whether the statement is true or false. Cellular repair of CRISPR-Cas9 mediated double-strand break by non-homologous end joining mechanism result, results in frameshift mutation. And the options are true or false. We can take a couple of minutes and then try to answer this.
So
Hmm.
non-homologous and joining.
Yeah.
So there is a nothing act as a template. No DNA strand acting as a template.
Hmm.
So so it is not possible for the DNA repair system to predict how many bases have been lost.
Okay.
It may add more bases or it may add less bases also. So that results in the frameshift mutation. That is the gene is knocked out.
Right. Yeah. So yeah. Yeah. Before going, Ignatius, do you have a difference, or would you also go with the same?
I know that the non-homologous end joining is associated with the indels at specific sites
Yeah, insertions or deletions.
and deletions.
Yeah.
But I'm not sure if they cause just point mutations or frame shift mutations. So,
Okay. So, huh. Okay. I can clarify that. So, rephrasing Surender, in non-homologous enjoining, we don't have any specific template, or we don't have patches of homologous regions with which a specific DNA can be altered, like we have in HDR. So, because of this, just to seal the double strand break, various numbers of insertions or deletions due to the proofreading activity and fidelity of the polymerase can occur, because of which it can affect in multiple ways. One is, if firstly we don't know the number of bases that have been lost during this double strand break. Let us say during this double strand break, three bases are lost. That would correspond to one particular amino acid given the ORF is at plus 1, meaning it's in the right coding, uh, how do I say, it's in the right set of codon. If it's in the start of the codon and three bases are lost, that would mean one amino acid is lost. But this would depend on how the further, or how the particular gene where it is being cut has the codon sequences set. So, because we don't really know about where it is being cut, and if it's being cut in the start of an amino acid or in the middle of an amino acid, which is like zero, if three bases are lost or four bases are lost, we can't really say if that would preserve mutations. So we can go ahead with saying that it can cause frame shift mutation. So in frame shift mutation, what exactly happens is the number of bases that are lost would lead to altering the ORFs. Like for example, let's say we have three bases, maybe 1, 2, 3, 4, 5, 6, 7, 8, 9. Let's use alphabets. Okay. And now let's say this is how it is initially present before the double strand break. And after the double strand break, let's say it leads to removal of this particular G and this particular C. So what would happen is, and or let's say this has been removed. Now what happens is, because of this one base loss, is there are no insertions or deletions. Or what would happen is, this would go here, this would come here. Similarly, what was initially started as ORF plus 1 is now shifted to plus 2. So you would have the entire set of amino acid sequence coding sequences downstream messed up from plus 1 to plus 2. That is a frame shift mutation. So this depends on the insertions and deletions, and in most cases, because this has been random, we can go ahead and say that this causes frameshift mutations. Was that clear, Ignatius, or should I try repeating
No, no,
that?
got it.
You got it? Okay,
Yeah, got it.
right. Huh. So it's true. It causes frameshift mutations. Huh. So the next question is various Cas genes are expressed and bind to option A CRISPR sequences, option B leader sequence, option C Cas array, and option D none of the above. We can take a couple of minutes and then answer this question. Yeah, does anyone want to answer? Yeah.
So Cas gene they bind to the CRISPR RNA, guide RNA. They bind to the guide RNA. So
Hmm.
also Cas bind to the virus DNA when virus DNA enters inside the bacteria. Then
Right.
Cas
It could
binds.
bind to either the virus DNA or RNA. Yeah, that depends
Yeah,
on
yeah.
the type
Or
classification.
Cas bind
Yeah,
or Cas binds to the guide RNA also.
right. Hmm.
So here uh, I find none of the above
None of
because
the
Cas
above.
is not binding to CRISPR, leader, and Cas array. They don't bind. None of the above.
Okay. Ignatius?
I just feel it's none of the above.
Okay. Let's check what the answer is. I'm sure that it doesn't bind to the leader sequence and the Cas array. So what I know is CRISPR array is the locus where you have all these the genes encoding for the Cas proteins, the leader sequence, repeat, and spacer. That is the CRISPR array. But I don't know what they mean by the Cas array. I don't know if they are mentioning about the genes that are, I mean, the array of genes encoding the Cas genes itself as the Cas array. So even if these proteins are being transcribed and translated, they would not bind to their own DNA. So it cannot bind to Cas array and leader sequence. And by CRISPR sequences, I'm not sure if they're talking about the repeat and spacer sequences. So let's see the answer. If by CRISPR sequence they mean the repeat and spacer sequence, it could mean that these Cas genes are expressed and bind to CRISPR sequences. But let's just check the answer. Yeah, so it's given as D, which is none of the above. So they don't mean that CRISPR sequence is the repeats and the spacers. Right. Hmm. Huh. So the next question is, what is the role of Q protein? Option A, it binds to DNA double-strand break ends and is required for non-homologous end joining in the TJ pathway of the DNA repair. It maintains genome integrity. Q directly and indirectly interacts with several C NHEJ factors and processing enzymes, serving as a scaffold for the entire DNA repair complex. Option D, only A. Option E, all of the above. We'll take a couple of minutes and then answer this question.
So, Q protein, actually, this protein is not found in the bacteria, but some of the bacteria they containing this protein, Ku protein,
Yeah, it is not present in E. coli, it's present in Bacillus, Streptococcus,
Mycobacterium.
Mycobacterium. Yeah.
It is
So,
required for the non-homologous end joining repair
right,
system.
right, right.
So, option A is correct. It binds to the double strand end breaks and required for the non-homologous end joining pathway for
Yeah,
DNA repair. If it is doing the DNA repair, so it is maintaining the integrity also,
genome
genome
integrity. Right.
and it direct and Ku also required one more protein. The name I am not remembering. That is also required for the non homologous end joining. So it recruits the protein. So it is acting as a scaffold. So the answer is all of the above.
Hmm.
So one
So
more protein is required along with Ku. Actually, I
it
don't remember.
requires another polymerase, as far as I remember, and one is called LIGD.
Linked.
LIGD.
Yes.
Yeah.
Yeah. Yeah.
So whenever they are using these systems in E. coli, they have this plasmid where it encodes for both the Q protein and the LIGD. So double strand repair can be achieved within E. coli. That's what they try. Yeah, what you mentioned is right. So this co-protein is required for non-homologous end joining, uh, which means that it can repair double-strand breaks. And when such a protein is capable of repairing double-strand breaks, it means that it is capable of maintaining the genome integrity, like you had mentioned, which also needs other factors like we discussed, which is LIGD. So all the options are right. Huh. The next question is D-Cas9 refers to option A, DNA bound to Cas9 protein, option B,
Uh,
catalytically, yeah, sorry. Huh.
Previous answer is D, na?
Option E, all of the above. Yeah,
E. Pradeva, yeah.
yeah, yeah,
Hmm.
yeah.
Okay.
Decas9 refers to option A, DNA-bound Cas9 protein, option B, catalytically deactivated Cas9, option C, Cas9 nuclease bound to guide RNA, option D, none of the above.
So it deactivated
It's
Cas9
deactivated. Yeah, do we have any
due to
other
some
information on that? Like what are the kind of mutations?
some
Any idea
mutations
on
are
that?
done.
Huh.
Which kind of mutation is done? I don't know, but some mutations are done in this Cas9. So it deactivated because of that mutation.
Right. Huh. So as far as I remember, two mutations were mentioned. One, so both of these mutations correspond to nucleus activity. And specifically, one histidine was mutated to alanine. The other mutation was also done so that it can be changed into alanine. So what happens with this DCAS systems is that they will still be able to bind to the guide RNA and sit on the DNA, but they will not be able to cleave the DNA of interest. So that is the DCAS9 or deactivated Cas9. So now this is done. What we can do further is discuss the right answers for week 6 course. Since we have about 20 minutes. Yeah, so in week 6, we were looking at metabolic flux analysis with labeled carbon. The first question is the difference between 13C MFA and FBA. And the options are metabolism-wide cofactor balancing is applicable in FBA but not in 13C MFA. Growth maximization assumption is a major criterion in FBA but not for 13C MFA. 13C MFA uses isotope labeled metabolites to constrain metabolic fluxes, but FBA does not. Option D, all of the above. So, what we need to remember is that in metabolic flux analysis, we only take a small portion of the network and give it an isotope labeled metabolite. Whereas in FBA, we think we assume that in steady state mass balance mass is balanced, and always in all the cases, unless we have knockouts leading to depletion of biomass, we assume that maximization of growth is the objective function. And under this assumption, we can also put in that the cofactors are balanced. So there is no constraint for lack of balance of cofactors in FBA. So the right answer is all of the above. The next question they ask about this logic of push and pull mechanism and identify the reactions that can be eliminated to maximize the production of metabolite B. This is similar to the question that we had solved. So to maximize B, B should not be converted to uh, D. So R5 should not be there, and A should not be converted to C. Full of A should go to B. So R3 should not be there. So R3, R4, R5, and R6. If these are removed, B will be maximally produced. The third question, consider an MFA experiment using glucose molecule labeled at first carbon or a mixture of uniformly labeled carbon. How many isotopomers can be found in a molecule consist of two carbon atoms? So we had discussed earlier, when we have two carbon atoms, the number of possible isotopomers could be a molecule with zero carbon atoms labeled, a molecule with one of the carbon atoms labeled, let's say first carbon atom, a molecule with the second carbon atom alone labeled, and a molecule with all the four carbon atoms labeled. These are the possible isotopomers. So this gives rise to four different combinations, which is the answer. And this can also be given by the relation of power n, 2 power n, where if you have two carbon atoms, you'll have four isotopomers. And the next question is the push-pull mechanism of metabolic engineering. So what they ask here is whether does it enhance stability of genetic modification? No, it does not. Does it increase the efficiency of transcription and translation? No, it does not. Does it improve the overall yield of the desired metabolites? Yes, it does. And minimize the impact of environmental fluctuations? No, it does not. So in the push-pull mechanism, what happens is you enforce such that precursor metabolites are produced in large quantities and it is directed towards production of your metabolite of interest. That is the push-pull mechanism. The next question is, which of the following statements best illustrates the difference between opt-knock and opt-force? So opt-knock focuses on identifying gene knockout strategies to maximize specific objective, while opt-force employs both gene knockouts and upregulation down-regulation strategies to achieve desired metabolic outcomes. Yeah, so that is the right answer. The next question is, in isotopomer distribution vector, a glucose molecule is represented 0, 0, 0, 0, 0, 1 bin. This implies that glucose molecule is labeled as a single 13 carbon isotope in the first position. Glucose molecule is completely unlabeled. Glucose molecule is labeled with a single 13 carbon isotope in the last position. None of the above. So what we have to understand here is 0 means absence of label, 1 means presence of label, and position of the label indicates the carbon atom. So here position of the label is at 6. So the sixth carbon atom is labeled, and that is the last carbon atom. The option is option C. Glucose molecule labeled as single 30 carbon isotope in the last position. The next question is consider carbon exchange reactions R1, where B plus C gives A, and choose the correct isotopomer mapping matrix for B to A. So we had performed a similar calculation. If needed, this can also be discussed as a part of the final session, which we will have after next week's week 8 uh, problem solving session, and which is not a biomass precursor.
So can I have
Yeah,
one question here?
yeah, sure.
Because now in this matrix, two or three cannot be possible. So somewhere
Because it
written
has
two, three.
because it has this three, is it? Or what exactly prompted you to say so?
Because if we are matching, we have to put zero or one. We have to put here. That's like if product is radio labeled and our reactant also radio labeled, then we put one. Otherwise zero. So it is my just opinion. I'm just
No,
discussing
I didn't get what you said. Can you repeat? The product is
because
radio labeled.
in option D, na D and C,
In option D and C,
three is written, three,
yeah,
two
three and
written.
two. So we that
So it
cannot
is
be
not
true.
possible here
Yeah,
in this matrix.
true. These two are not right.
Okay.
Even B isn't right because you have this entire column
Yeah. Yeah.
having ones,
Yeah. Yeah.
but we need to know how to do this. We also performed a similar calculation with a particular order in our mind. Although the order does not matter, since the answer has a specific order of ones and zeros, the way in which we solved the order that we had given was not right. But we can discuss this again if we need in the future
Yeah. Yeah.
problem solving sessions. Yeah. Which of the following is not a biomass precursor? E- huh, 2-phosphoglycerate. We had a discussion on the same the other day, and the solver used to optimize a small network is linprog. And about opt-force, an optimization procedure for identifying genetic manipulations leading to targeted overproduction. Opt-force is used for gene annotation. It's used for reconstruction of dynamic models and of the above. So all the three answers, three options B, C, D are wrong. Only option A is right. Yeah. So with this, we can conclude for this session. If there are any specific questions or topics that you want to be discussed, you can either post it in the Google groups so that it'll be we can spend time on specific problems rather than unimportant stuff. Although we'll be giving an overview of what all was covered in the course. Is that okay?
Okay. So,
Yeah. Thank you, Surendra. Thank you, Ignatius.
thank you.
We can.
So next week will
Yeah.
be the last
Next
class.
Monday,
Last.
we'll have the last tutorial session. Mostly on
Okay.
Tuesday, I think you will be receiving an email soon. On Tuesday from 6 to 8, we'll be having another session where we will have this revision. And if you have any doubts, we can clarify that.
Okay. Monday plus Tuesday.
Yeah,
Monday
no,
plus.
Monday's class will be for week 8's lectures. That
Okay.
is
Okay. Okay.
separate. Apart from that,
Okay.
on Tuesday, we will have one session.
Okay. Okay. Okay.
Right, right. Thank you.
Can we leave?
Yeah,
Thank
yeah,
you.
sure.